Hello everyone and welcome to the Altasciences webinars and podcast channel. Today in our consultant series, where globally recognized consultants discuss how they are addressing some of the most pressing drug development topics, we are excited to welcome our special guest, Dr. Brian Short.
Dr. Short is the president of Brian Short Consulting LLC, a toxicologist and a board-certified veterinary pathologist with over 25 years of experience in non-clinical ocular drug development of small molecules and biologics, and more recently, gene and cell therapies. Dr. Short has contributed to the successful development of numerous approved ocular drugs in his position as a senior director of toxicology and then pathology in his 17 years career with Allergan. As an independent consultant for the past 7 years, he has consulted for over 100 ocular projects and interacts regularly with FDA Ophthalmic Division on behalf of sponsors company. He has recently served as counselor for Roundtable of Toxicology Consultants and is a member of STP's Special Science International Harmonization of Nomenclature and Diagnostic Criteria for No Rodents. Dr. Short is a leader in the ocular industry space, and he will share some insights about ophthalmic toxicology and the challenges and opportunity in non-clinical ocular development.
Brian, thank you so much for being with us today and sharing your ophthalmic toxicology expertise. Would you mind telling our audience how you got into drug development?
Sure, Norbert. First of all, I just want to thank you and Simone and the rest of the Altasciences group for inviting me to talk about this subject, because I'm very passionate about. As far as how I got into drug development, my research background was in environmental toxicology and carcinogenesis. This was after I was a veterinarian with a background in pathology, and I studied the kidney actually first, alpha-2 nephropathy and renal carcinogenesis. I was a postdoc at the Chemical Industry Institute of Toxicology in Research Triangle Park, Carolina, and I was there doing this concurrently with my graduate studies at University of North Carolina in Chapel Hill. I was interested in pharmacology, and I went to a large pharmaceutical company after I graduated my PhD, SmithKline Beckman at the time in King of Prussia, Pennsylvania. It became SmithKline Beecham, and then it became GSK shortly after I left, 10 years after being there. It seemed like a great place. It was a good fit for pathologists. We were also considered toxicologists and not just slide readers. We had some study directing experience. I was involved as a project representative, everything from short-term tox studies to and there were safety pharmacology, reproductive and developmental studies, and carcinogenicity studies, all done in-house. So that was a great experience. But then after 10 years, I decided to step up and take a leadership role, and I was recruited to go to Allergan in Irvine, Southern California. Now, I had very little prior interest or experience in the eye, other than broad training as a pathologist. So this was a steep learning curve for me with ocular drugs. There were also dermal drugs at Allergan, and at that time, a not so well-known drug, neurologic drug that Allergan had, which was actually for injecting around the eye, called Botox. And we all know the Botox success story after that, but we worked a lot of medical therapies there at Allergan, such as urinary incontinence and other systemic or peripheral nervous system diseases, which required animal models for safety. For ocular experience, I was surrounded by many capable scientists and technicians with extensive ocular drug development experience and running ocular studies.
Great, Brian. So my next question would be, what would you say are maybe the 3 most important things to know when developing an ocular toxicology study?
That's a very good question, because sometimes companies that come to me haven't thought about some of these things before they want to get ready to do toxicology studies. But the first thing I would ask them that, based on what they're trying to develop this molecule, is it a reformulation of a previously approved drug, either approved by the ocular route or a systemic route, or is it what we call a new chemical or biologic entity that really hasn't been into people before? And there's different categories, as we know, of drugs as well. We have, small molecules and biotherapeutics, and then we have peptides. And I consider a peptide a small molecule if it's less than 40 amino acids long, or if it's greater than 40 amino acids, it's considered a biotherapeutic. This is a recent FDA classification. Or is it a oligonucleotide, which in some senses is considered a small molecule with potential off-target effects, or also is considered a biotherapeutic because it can be immunogenic. And then, of course, there's other categories such as gene and cell therapy. So that's the first question, just to try to understand what is the molecule and what is regulatory history. And the second is what's known about the formulation, because there has to be some formulation development. We don't want to be using formulations that are going to change a lot before we get to the clinic. And that can take some work to know if the formulation is viable to test. And then what do we know about ocular, what are called pharmacodynamics or pharmacokinetics of the molecule? It's very important to know what the scientists in the company think. How long will this molecule be biologically active in the eye? Or what is the duration of the drug in the eye? How long does that last? Is very important. And then the last is, what about the clinical plan? Is there a target, product profile for this drug where they know what is the intended dose regimen? What will be the concentration range they would want to get to the eye? What will be the volume they want to administer? And how often do they want to give it? Because all the toxicology studies really need to closely mimic what the clinical dose regimen will be. So that's my answer of the three most important things to know when you're developing an ocular toxicology study.
How important is it, in your opinion, as a consultant, to be familiar with all the procedures involved in the study design?
Well, it helps to be informed on what are the appropriate study procedures, based on the route of injection or administration and the type of the molecule, as I discussed previously. A person doesn't have to be intimately familiar with everything because Altasciences has experts such as people like Simone that know a lot about a lot of the specialized procedures that happen with ocular toxicology studies. But I think, as we know, the sponsors need to, ask Altasciences, what kind of a study they think they need to run. And I think it's really best practice for a consultant and the sponsor to work together and provide a short 1 to 2 page protocol outline for each study, with a short objective, what the rationale is for the dose selection, and a study design table, and then a table of what I call parameters of everything that the company would like Altasciences to look at and how often they would like to evaluate these parameters. I think that's the first step for the company to understand what do they think would be a good idea. And then go to Altasciences and I think ideally have a talk with them and with the scientists that you have on staff, such as your study directors and Simone, to see if they're all on the same wavelength about what would be, a good parameters to look for. It can be very different from a dose range study to a GLP study about what would be needed. So I would say it's important to be really familiar with the standard ocular examination techniques used in ocular tox studies, including there's ocular observations, which is the Dre's technique, which has a scoring very well known over decades. It's usually conducted by technicians. There's, of course, the ophthalmic examination, which is usually slit lamp biomicroscopy, indirect fundoscopy, and for most studies by a board certified veterinary ophthalmologist such as Simone. And then there's other more specialized techniques, as I said, depending on the type of molecule, the route of administration. But I don't feel everyone has to be intimately familiar with all these techniques. That's why, the experts are there and covered to execute these and also give advice on, what techniques are needed. But I'll go over some of these techniques. Pachymetry, which is for corneal thickness, that's usually for a topical ocular study. Specular microscopy is for evaluate the corneal endothelial cells. There's gonioscopy, which looks at the angle of the anterior chamber, or you could look at anterior segment optical coherence tomography, known as OCT. And that would be if you're putting an implant drug into the anterior chamber, which is called the intracameral space. Then there's another category of more for evaluating the back of the eye, things like electroretinography, fundus photography, fluorescein angiography, posterior segment OCT, and another technique called confocal scanning laser ophthalmoscopy, CSLO. These are mainly for intraocular studies, most common intravitreal, but also subretinal or suprachoroidal routes. As I mentioned, the resident ophthalmologist, Simone, has a good working knowledge of these specialized procedures and can help determine which of these would be good to include based on the study objectives.
So the next question would be, what would you recommend to our audience to anticipate the different challenges faced when choosing the right animal model for an ocular toxicology study?
Oh, this is a very good question, and it deserves a lot of consideration and thought, especially in this day and age with the 3 R's of trying to use the right species and reduce and refine the species that we use. And it does get back to the earlier question, and my response to that the right animal model depends on determining if the drug is a reformulation of a previously approved drug, or is it a new chemical or biological entity, or is it perhaps maybe a gene or cell therapy product, and also what is the route of the delivery. Now I'm going to kind of go in sort of a stepwise approach and just give you some of my thoughts when thinking about some of the animal models based on some of these categories. I'm going to try to go from probably the most simplest to the most complex. And the simplest would be for a small molecule, a reformulation of a previously approved ocular drug, or what's known in FDA as a 505(b)(2). Generally, rabbits are sufficient as a standalone species by any route of administration. The next level of difficulty would be a small molecule reformulation of a previously approved systemic drug, also a 505(b)(2). Now, so that means that the drug has been approved systemically, but has not been approved by the ocular route. So in this case, generally, my experience is that the FDA expects to see 2 ocular species, usually including rabbits as sort of the default species. However, sometimes rabbits may be the only species that you do an ocular toxicity study with this situation. And it can be adequate as a standalone species if you can show that the previous systemic route studies had higher ocular exposure than given by this ocular route. So what a company would do is that they would do what I call an ocular and systemic route PK bridging study, where you give both routes to that animal who had already had an extensive systemic study, and you'd look at the eye by both routes and answer the question, is there higher ocular exposure from the systemic route? And sometimes there is, because the doses that we give to systemically can be large and they can get into the eye, whereas the doses that we give to the eye are really generally small. The next topic I'd like to go to here about species considerations is, what are the second species non-rodents for small molecules if we do have to pick a second species? And this is mainly, I'm covering right now, small molecules. And the first order is that, or the first recommendation is that monkeys should not be used for small molecule drug development. And this has been a recent guidance that the FDA issued earlier this year. This was due to the pandemic and the reduction in supply of primates and the increased urgency for their use in some COVID research and development activities. So another species should be used instead of a monkey for small molecules, even though we all recognize that monkeys' eyes are more similar to humans than some of the other species, but the other species are acceptable. Dogs, for example, they are considered adequate, for topical ocular. Their intracameral studies, their angle in the eye is most like human than other species. Also, they've been used for intravitreal studies as well. All the triamcinolone corticosteroids were developed in dogs with intravitreal studies. Mini pigs are becoming used more and more, and they also considered adequate. They're mainly adequate for intraocular studies, intravitreal studies, subretinal studies. They could be a little more difficult for topical ocular, just due to some of the manipulation with eye drops several times a day in a large animal. Then the next category I want to talk about is for novel small molecules, small peptides, oligonucleotides, and also biotherapeutics. So these are new chemical or biologic entities known as a 505(b)(1). So for this, the FDA usually would like to see 2 species for ocular toxicology studies. They're generally expected as long as they're pharmacologically relevant for the biotherapeutic. They don't have to be pharmacologically relevant for the small molecule. Now, rabbits can be used even if they're not pharmacologically relevant. They can be used for dose range studies for the single dose of any route. And they can be used for GLP studies if they're pharmacologically relevant and if immunogenicity is not an issue. As we know, sometimes rabbits, when they're injected with a biotherapeutic several times, they will mount a strong immune response, which may prevent them being considered an adequate species. Monkeys are usually pharmacologically relevant, and therefore they are justified in most cases for biotherapeutics and some gene therapies, but not usually cell therapies. But I think everyone now in this day and age is always looking to see, well, yes, we can use a monkey, but what are the other reasons why we can't use, say, mini pigs or dogs? And even I think the agency is looking for companies to explore either on an in vitro basis, genetic homology, gene amino acid homology, if mini pigs and dogs are also relevant and could be considered as alternative species. And they can be, as I said before, they can be considered for intravitreal, subretinal biotherapeutics, gene and cell therapies, based on pharmacological relevance. So that's pretty much in a nutshell, all the different species and some of the considerations that we have to give to all of them.
With your experience working in the industry, what are the top 3 things you look for in a preclinical contract research organization and or a contract drug manufacturing organization partners?
Oh, well, first I want to talk about the contract manufacturing partners. I personally don't have a lot of direct experience in the role in the manufacturing groups, as from a consultant, but I certainly indirectly interact with the contract manufacturing companies through usually a consultant who is familiar or an expert in this area of formulations. I think there are important things from my perspective, for an ocular drug contract manufacturing, and to make sure that they understand what are some of the limits that are necessary, especially for injecting the drug into the eye for the impurities. Things like endotoxins, host cell proteins. The eye is generally more sensitive than giving the same drug by a parental route. So the company needs to know that some of the specifications need to be lower. There are certain acceptable limits there that I usually weigh in on when the contract manufacturing group is, developing their formulation. But with most of my experience with the contract research organizations, the test facility, so the top three things would be, what level of experience does the contract lab have in conducting ocular tox studies? If I'm not familiar with the contract lab, I will generally ask for like a summary for like the past 3 years of, how many ocular toxicity studies have been conducted, what's been the species, the route, the study duration, and the GLP status. So that's the first thing. The second thing that's important to me is, who is conducting the ophthalmic examinations? Are they a resident ophthalmologist? Are they, nearby and come in on a contractual basis, based on the testing schedule? And what specialized procedures are they capable of conducting? Do they have the equipment? Do they have the experience to conduct some of these procedures that we talked about earlier today? Fortunately, Simone at Altasciences here in Scranton, Pennsylvania, is a resident ophthalmologist, which is a real plus for that facility. And then this last thing would be to me is that who conducts the pathology evaluation? Is it in-house or a contract pathology CRO? And what experience do they have with ocular pathology? I mean, both are good, but I like to know who are the people, what is their CV? Do they have any experience in this area? I would want to say that there's been some recent activity with ocular pathology. The FDA 3 or 4 years ago was actually asking for serial sections of the centralis of the retina in different species, or the macula. And I think we went through with the Society of Toxicology Pathologists and put out a publication to standardize the approach to ocular pathology, which is now usually 3 to 5 sections, step sections through the eye, to make sure that we're looking at all the structures and getting an adequate evaluation of the eye for ocular toxicology studies. And I think most of the laboratories now are familiar with some of these more specialized histology techniques that are not usually done for other route studies outside of the eye. I do know that Altasciences contracts with Stage Bio, a large CRO, and they do have expertise in ocular histology and pathologists there who are expert in ocular histopathology. And that are the 3 top things that I look for in CRO partners.
Thank you, Brian. These are very great answers. So to conclude the interview, I would like to ask our 3-question feature for you. So the first one would be, what do you like to do outside work?
Well, I usually like to be active for 1 to 2 hours in the morning. where I live here in Laguna Beach, I'm surrounded by nature and scenic views. I live about 2 miles from the coast, up at the top of a ridge, about 1,000 feet above sea level, in a house that overlooks not the ocean, but a regional park behind the ridge. And it's a canyon. There's a lot of hiking and biking trails, community park with tennis and pickleball courts that overlook both the ocean and the Southern California mountains. There's plenty of hilly neighborhood running routes and a community pool for lap swimming. So I have a lot of recreational choices every day to choose from.
The second is, can you tell us something we can learn about you from reading from your resume?
The past few years, I've gotten more involved in ocular gene and cell therapies, and I am enjoying learning these new technologies and how to design and interpret studies, biodistribution, and also the interchange with the FDA. There's a different group there that regulates this category, or the Office of Tissues and Advanced Therapies, otherwise known as OTAT. And so it's been a really just enjoyable, just a new challenge that I can't claim that I have a lot of experience in the last few years, but am gaining it and enthusiastic about the very promising field and the advances. As we know, the first gene therapy that's been approved has been for the eye. So that's exciting to me.
Awesome. And finally, can you let the audience know a fun fact about yourself?
I enjoy visiting national parks and wineries in the Western US with my spouse, not necessarily in that order, but that's the first thing. There's a small other thing. I have 4 children, adult children. They're all between the ages of 29 and 32. So weddings and grandkids are in my near future, although not necessarily in that order. But that's the other fun fact about myself.
Well, thank you so much, Brian, for sharing your great expertise, knowledge, experience, and being here with us today. And thank you all for listening. Be sure to check out our other podcasts, webinars, and videos, and the Altasciences webinars and podcast channel.
Thank you so much, Brian, for being part of this podcast, and we look forward to more of the same in the future.
Thank you for having me, Simone and Norbert.